So the turning point for data integration came with the multi-nearest neighbors method paper in Nature Methods, I think, or Nature Biotechnology. And that was the turning point in how the methods changed their approach, not only for matrix multiplication directly to a graph-based integration. So how does MNN work? And MNN stands for mutual nearest neighbors. If you know what shares near neighbors, and we'll also talk about that later. But basically what MNN will do a PCA in one batch of the data, and then project the cells using PCA from all the other batches to the same PCA as the one batch that you're comparing to. So all of them are kind of aligned, the data sets in their space, as is shown in here in figure A. And then what we want to find is the K and N, as I mentioned in the dimensionality reduction lecture, find the nearest cells between the data sets, not within the data sets. But now I want to find what are the nearest neighbors of this cell in the other data set. And then these neighbors here will be paired, for example, and some of those cells will not have a matching pair here, but that is okay, because now based on these pairs that we see here, we can find what are the vectors that are necessary for one batch to merge in the other one. So for example, like if you have all those vectors here, we can find, oh, we need to reduce the amount of in these coordinate so that these two clusters merge with these two. So these are basically what these vectors mean. It's like, how much should I change this cell to match the other data set? And that these vectors can be just extended to the other cells that don't have a match here as well. In that way, you can have like all those data set combined, data set 1 and 2, and then you can add a third batch and then do that iteratively. So for example, like the third batch only matches this cell type, and then these also will be corrected to that space. And the good thing about this is that it's very fast, because it only accounts for the K and N, so like only the nearest neighbors, and not the whole data set as for the matrix factorization. So it is one of the turning point for data integration. But it also has some assumption that you need at least one cell type that is present in both data sets. So if you have completely different data sets, you will try to merge them anyways. So you need to take care of that also and think about it. So yeah, some of those things are a little bit more technical for now. And you need to think that if the batch effect variation is much smaller than the biological effect, you might join things that are not the same. So yeah, so in many cases, so for example, here you see an uncorrected data set. Here you see the MNN, which is a corrected data set. But if you do, for example, Lima or Combat, which are the regression-based methods, you see that they don't correct for the batch at all. So this is one of the advantages of the MNN, because then it adjusts for these local variances here. Another method that is used now in Seurat version 3.
Can I have another question?
Yes, absolutely.
So you're saying that at least one cell population has to be present in both batches, but I presume you could also have 5 or 6 batches or more, or would you have to do one at a time? So would it also work if you had kind of like a bridging cell population that are present in some of the samples, but not all, but at least one cell population is present in at least 2 of the batches? Do you understand what I mean?
Yes, exactly. So imagine that you have this batch 1 with the purple, red, and blue, and then you try to integrate with batch 3. So that will cause either an error, because not an error, you will integrate it for you, but maybe the cells from batch 3 will be present in all those 3 here, because there is no respective yellow cluster here, right? So you will try to find the integration, but the integration will not be correct.
Yeah.
So, and one way to visualize it is that if you run a TSNE of batch 1 and batch 2, or batch 3 in this case, you'll see one cluster and 3 clusters. And after integration, you see that this cluster from one batch is spread all over. So this is a problem later. And I will come to that later on as well.
Yeah.
So in that case, you need, and most of those algorithms will do the batch iteratively based on that. So you'll find batch 1 and batch 2, and then you have a cell type that is only present on batch 2 that can be used to match the batch 3.
Okay, yeah. All right, thank you.
I should mention that these MNN, you can specify the order you supply the data sets in. So if you would have the wrong order, you would mess up this integration completely if you did 1, 3, 2 instead.
Yeah.
It automatically does it by size, right? But that might not be what's best, or is it how they're provided?
I thought it was by the number of matching nearest neighbors.
Okay, maybe they update. Before it was by size, but yeah, we can look at that later.
Yeah, and I think the Seurat and CCA will do, I think is also by the number of integration anchors. Anyways, so the Seurat, instead of using PCA plus the graph, you will basically use CCA plus anchors. So I actually don't have the graph on explaining CCA here, but CCA, in comparison to principal component analysis, CCA stands for canonical correlation analysis, and it basically captures the correlated sources of variation between two data sets. So if two data sets vary, have some variation in a let's say in a couple of genes, what you want CCA to do is to find which genes are well correlated in one data set and are also well correlated in another data set. And because of that, then you can basically try to align the data sets in this way. However, the batch differences are still a little bit present here. So CCA does not correct for everything. And what you can do is then, exactly as the MNN, find the corresponding cell across the data sets, trying to find the mutual nearest neighbors, and Seurat uses actually MNN in the background. Then compute the data adjustment or calculate the vectors to correspond, adjust the data, and then apply the adjustment for the data set. And then this, in a way, visualized. So we have CCA. In some cases, you will not. You will basically just align the data sets, but then you need just to do L2 transformation, which is an adjustment of the data set, so it corresponds to one another. And basically, then find the anchors using mutual nearest neighbors, which is exactly the same as for the MNN. So you try to find which cells correspond to the other ones. And in some cases, you'll see, for example, that some cells are not actually present in the reference data set. And then those cells should be another cluster separately. And as well as for the MNN, it also performs very well. The only problem with the CCA is that since you want to you don't do PCA, you do CCA before, and CCA takes a long time to run. And usually that's why the Seurat method is quite good for data integration, very, very good. But it might take some more time than the fast MNN, because it both computes corrected matrix of gene expression and needs to compute CCA before integration. In the Seurat object, so you'll see that there is another object created. It's not RNA in the assay anymore. Then it becomes something different. It becomes CCA. So another method, which is also similar to MNN, is called Scanorama, and that is present in Python. Combat is also in Python. And I don't know which one we have more in Python. The MNN is also in Python. And Scanorama was originally developed in Python, but there is the R implementation as well. So it's virtually an implementation of fast MNN. But now, instead of using PCA, it uses singular value decomposition, which is the general idea of PCA, but PCA is a subset of this method. And you try to find, again, the key nearest neighbors across data sets, and then compute the merging vectors. But the idea of Scanorama, it optimizes for finding the best way to integrate the data sets like this. The idea of this Scanorama in principle is also to find matching points between images. So Scanorama can be used also in images for joint integration, and created like this panorama. So if you have several pictures, you can create one big image with that. So it uses some of those measurements for finding which are the best cell types instead of the mutual nearest neighbors. It tries to find it in a simple, different way. But essentially, is exactly the same thing as the MNN. So finds pairs of mutual neighbors across data sets, and then try to create a panorama of all of them. But instead, the only big difference is that instead of doing that per batch, it accounts for the information across all the data sets at once. And that speeds up a lot of the computation and avoids this problem in like which order of the data sets you are trying to merge with. Another thing that I can just briefly mention, I don't have a slide on harmony, but harmony, it's also virtually the same thing as Scanorama, tries to find everything at once, but you will just cluster your cells beforehand and then try to find a vector for correction of the data set using the cluster information.
Does that affect?
Sorry, yes?
Does that affect the reproducibility, or do you get still the same clusters then each time? Or?
Which methods are you comparing? You mentioned
the Scanorama, because you said you do the clusters first. If it affects like the
yeah, exactly. So the harmony will do clustering in each individual data set before, and then try to see which clusters are present in each data set, not per cell, but per cluster. Yeah. Which is essentially will give similar results. If you're running the Seurat package or the SCRAN, you will see the results. They are very similar as well. And also for the MNN and Scanorama, you'll see the results are pretty much similar as well. But the good advantage of Scanorama, for example, and harmony as well, is that they are not much memory heavy. So they are really good to use on our local your personal computer. And they scale well for millions of cells. And both of them are really, really good and fast. Although CCAs, they say that it's very heavy in memory, and that is completely true. MNN, here they are using not the fast version of MNN, but MNN is also pretty fast.